Hi, this is Dr. Jessica Han. In this section, we're going to talk about energy conversion and the function of enzymes. All the living organisms require energy. For example, a hummingbird needs energy to maintain prolonged flight. The bird obtains its energy from taking in food and transforming the energy containing in food molecules into forms of energy to power its flight through a series of biochemical reactions. Most life forms obtain energy derived from the sun. Producers, such as plants, use photosynthesis to capture energy from sunlight and convert it to sugar molecule glucose. Consumers, like animals, get energy by consuming the producers or other organisms. Energy can be converted through chemical reactions. Catabolic pathways are those that generate energy by breaking down large molecules to smaller molecules. The body can not only break down large molecules, but it also have capacity to synthesize molecules. So reverse of catabolic reaction, there is an anabolic reaction. This reaction or pathway are being used by small molecules. They will be linked together to build into a large molecule. Energy is defined as the ability to do the work. There are two main types of energy. One is potential energy and the other one is kinetic energy. Many forms of each type of energy exist. Energy can be converted from one form to another. Laws of thermodynamics describe energy conversions. The first law states that energy can be converted from one form to another. The second law states that with each energy conversion, some energy is lost to the surroundings as heat. The example of potential energy and the kinetic energy can be illustrated by water. The still water has potential energy. Moving water, such as in a waterfall or a rapid flowing river, has kinetic energy. Energy can be converted as stated by the law. The food we consume provides our cells with energy required to carry out bodily functions. Light energy provides plants with the means to create the chemical energy they need. Energy can be converted through chemical reactions. Chemical reactions can either release or absorb energy. Delta G is a measure of usable energy. If delta G is negative, energy is released from that chemical reaction. The reaction is exergonic and will occur spontaneously. But still, it requires activation energy to get the chemical reaction going. If the delta G is positive, energy is absorbed through the chemical reaction. The reaction is endergonic and non-spontaneous. Enzyme is a protein. Enzymes can speed up the chemical reaction. It can lower the activation energy of the reaction, but the enzyme does not change the free energy of the reaction. Enzyme can be reused in other reactions. As illustrated in the diagram on the right, you will see the reactants originally need a high activation energy to start the chemical reaction to form the products, as shown in the purple line. With the catalyst from the enzyme, the activation energy can be much lower, as shown in the right line here, and still produce the same product. While enzyme performs its functions, it requires a substrate. Substrates are chemical reactants that enzyme binds. Enzyme has active site. Active site is the location where substrate fits. Once the substrate bind to the active site, the chemical reaction happen, then the reactants will be broken down into product, then being released from the active site. The function of enzyme can be in many ways. They can bring substrates into close proximity to react with one another, or it can create a beneficial chemical environment for reaction to occur, or it can bind many compromise bond structure and allow bond to break. This diagram shows the binding of the substrate with the enzyme through the active site. Then the binding will catalyze the reaction to form products, then being released from the active site. Then the enzyme remain non-consumed. Enzyme activity can be controlled. They can be either inhibited or activated. There are two types of inhibition. One is called competitive inhibition. In this type of inhibition, inhibitors will bind to the active site and block the binding of substrate. Another type of inhibition is called non-competitive inhibition. This is also called allosteric inhibition. The inhibitor will bind to the enzyme in a region that is distinct from the active site. The binding will cause the enzyme to change the shape or structure. The change of the shape will block the binding of the substrate. The enzyme can be activated too. The activator binds the enzyme in a region distinct from the active site and promotes the substrate binding. The figure here shows the allosteric inhibition. The inhibitor will bind to the enzyme, change the shape of active site. Therefore, the substrate cannot bind to the enzyme anymore. The activation will go to the same mechanisms. Activator will bind to the enzyme to make the active site to be more suitable to the substrate, then promotes the binding. Some enzymes require non-protein helpers to work. Helpers may bind temporarily or permanently to the enzyme to promote optimal shape and function. The example will be allosteric activator we just talked about in the previous slide. Cofactors are also required sometimes for certain enzymes. These cofactors are inorganic ions such as iron or magnesium. Some enzymes require other proteins called coenzymes to help with their functions. Those coenzymes are often organic, which means they contain carbon in their structure. Examples of coenzymes can be vitamins, vitamin B. The chemical reaction will not always go on and on. There's a mechanism to stop the reaction. This mechanism is called feedback inhibition. Feedback inhibition, where the end product of the pathway inhibits an upstream process. It is an important regulatory mechanism in the cell. For example, the first substrate combines to the enzyme, then produce a product. Then this purple product is called the intermediate substrate. He will bind to another enzyme, then produce another product. Then the reaction keep going. Once they produce into the end product will come back, bind to the first enzyme. This end product functions as an allosteric inhibitor. Then this inhibitor will change the shape of the active site. Then the substrate can no longer bind. Then the reaction will stop here. In summary, energy can be converted through chemical reactions. Enzyme is a protein that can speed up the chemical reactions. It can be reused. It can be controlled or regulated. The enzyme activity can be inhibited by inhibitors or activated by activators. Some enzymes require factors to help with their functions. Those factors could be minerals such as iron, which are called cofactors, or they can be organic factors, which are called coenzymes. This will summarize the mini lecture for energy conversion and enzyme functions.